Hello, everybody. Can you hear me?
Yes. We can hear you.
And you can see me, right?
Yes.
Good.
Yes, we can hear you.
And you can see the slides also. So we're all set. Okay. So I hope you had a good long weekend. And today we're going to start with chemistry for a few lectures, just to review some concepts. So we'll start with very simple, the atom. So what is an atom? An atom is made up of three different types of particles. The protons that are positively charged and the neutrons that are in the nucleus. So these two particles are inside the nucleus. Then on the outside, floating around in waves, are the electrons, and they are negatively charged. And there's only a certain number of electrons that are present in different what we call shells. So the first shell that is the closest to the nucleus of the atom can only hold 2 electrons. And this is a rule. Then the next shell can hold up to 8 electrons. Here you only see 1, 2, 3, 4, 5. But this atom could hold up to 8 electrons on the second shell. And as you go on moving from shell to shell, there's more and more electrons. The third shell can hold up to 18 electrons. So the outer shell of an atom is called the valence. So in this atom, the valence, which is the second shell, contains 5 electrons. So one good news, let me just say, is that everybody can see everybody's chat. So this is good. So coming back to the slide, this atom, which is an imaginary atom, contains 1, 2, 3, 4, 5, 1, 2, 3, 4, 5, 6, 7 protons, should contain 7 neutrons, and it has 7 electrons. 2 in the first shell and 5 in the outer shell. So you would say that this atom is neutral. Why is it neutral? Because it contains 7 electrons that are negatively charged and 7 protons that are positively charged. So atoms that are neutral cannot form molecules because the atoms cannot attract each other. But there are certain atoms that become ions. When they become ions, they become ionized, and we will discuss this a little bit later. That means that the number of electrons and the number of protons is different. But now let's keep this simple. This is a neutral atom because the number of protons, in this case 7, is the same as the number of electrons, which is 7. And the shell, the first shell where the electrons are floating, can only hold 2 electrons. And the second shell, which in this case is known as the valence, contains 5 electrons, but it could contain up to 8 electrons. Okay, now let's see how can you define atoms in terms of number of protons and so on. So the first parameter that defines an atom is the atomic weight. So it's logic that the weight of an atom equals the sum of the mass of protons, the mass of neutrons, and the mass of electrons. Now, the mass of electrons is minor because electrons are very small. So when you add up the mass of protons, neutrons, and electrons, then you can calculate the atomic weight. So the weight of an electron, of an atom, sorry. And the unit of the atomic weight is Dalton. A Dalton is close to the weight of a single proton. So hydrogen contains only one single proton. So the Dalton could be similar to the weight of hydrogen atom because it contains only one proton. Now, let's see the second parameter or number that defines an atom, and that's the atomic number. So the atomic number, you have to remember, equals the number of protons. So remember, protons are positively charged, and the atomic number equals the number of protons. So if in a neutral atom, we know the number of protons, because this atom is neutral, you could calculate the number of electrons. So the atomic number, the number of protons equals the number of electrons in a neutral atom. So if I would say, let's say oxygen has 8 protons, that would mean that the neutral atom of oxygen would contain 8 electrons. So that the atomic number of a neutral atom indicates also the number of electrons. Now, what's the mass number? So the mass number is different from the atomic number. Atomic number is only the number of protons in the nucleus. Now, the mass number equals the number of protons and neutrons. So if the mass number of sodium is 23, then that means that it contains 11 protons and 12 neutrons. Oxygen 16 contains 8 protons and 8 neutrons. Now, let's make this thing a little bit more complicated. An atom, the number of protons in an atom is always the same. Let's say oxygen has 8 protons. So the atom of oxygen always contains 8 protons. But the number of neutrons can vary. That means, as you can see here, that this atom of oxygen, oxygen 16, contains 8 protons and 8 neutrons. But oxygen 17 contains 8 protons, but now 9 neutrons. Oxygen 18 contains 8 protons, but now 10 neutrons. So you can see here that the atom of oxygen can exist as O16, O17, and O18. But these 3 atoms of oxygen, they all contain the same number of protons, but it can contain different number of neutrons. This is important when we are going to soon talk about isotopes that are atoms that are radioactive. So remember this, that the mass number of an atom is always the same. What can vary is the number of neutrons. Okay, so very intelligent scientist known as Mendeleev was able to arrange elements in what we call the periodic table, a periodic table. So what is an element and what is an atom? So an atom is the smallest unit of an element. So an element is composed only of one type of atom. So it seems that atom and element could be interchangeable, but they're not. So this table is a periodic table that lists all the elements that exist. There are about 180 elements. And Mendeleev was able to arrange the elements according to their atomic number. Remember, atomic number is the number of protons. So that's what the numbers here mean. So horizontally or in rows, the elements are arranged by atomic number. So you see 1, 2, and then you go 3, 4, 5, 6, 7, 8, all 1, 2, 3, 4, 5, 6, 7, 8, 9. So these numbers represent the atomic number of the element. So they are arranged in rows. But then Mendeleev was able to arrange these elements also in columns. And what do the columns mean? The columns mean that these elements in each column have similar properties. So this seems like a very simple idea. You just arrange the elements based on the number of protons, and then in columns arrange them in with similar properties. But this was quite hard to come up with this idea, if you think about it. And what's the use of this periodic table? So this allows scientists to look at one specific element and immediately know how many protons it has and what are other elements that have similar properties. So this is a very simple kind of arrangement of elements, but that can be very useful information. Okay, so let's look at our body. That's what we are interested in this class. So our body, 96% of our body is made up of 4 elements, only 4 elements. So 4 elements out of these 180 make up our body: carbon, nitrogen, oxygen, and hydrogen. So our body is very simple in terms of element component. We only have 4 elements that are brought together to form all kinds of molecules that then go to organelles and organs and cells and so on. But made up our whole body is made up, 96% of our whole body is made up of these 4 elements. And if you add calcium and phosphorus, then we go up to 98.5% of our body weight. So you could say, well, our body is very simple, but how complex it is with all these physiological aspects, and also the intellectual aspects of our body. It's only dependent, 96% of it is contains these 4 elements. Okay, now let's move on to isotopes. And why am I explaining to you isotopes? Because we take advantage of isotopes to generate diagnostic tools, such as PET scans. Remember, we talked about PET scans in last lecture, how PET scans can be used to determine the function of certain organs. And it depends on a radioactive isotope. So what are isotopes? So isotopes are atoms that contain the same number of protons. Remember, the number of protons is specific for each atom. So it contains the same number of protons, but contains different number of neutrons. So remember, I gave you the example of oxygen. Here you have oxygen that contains different atoms of oxygen. They all contain 8 protons, but the number of neutrons varies. Okay, now when we talk about isotopes, isotopes are 2 atoms that contain the same number of protons. So let's say here carbon. Carbon has 6 protons. That is its identity. It contains 6 protons. But the number of neutrons can vary. Remember, this number here represents the atomic number. So the atomic number is the number of protons and neutrons. That's the mass number, sorry. This number here represents the mass number, which is the number of protons plus the number of neutrons. So we go back to the carbon here, that's the mass number. And in this carbon 12, we say carbon 12, it contains 6 protons and 6 neutrons. But this is carbon 13. It still contains 6 protons, but its mass number is 13, so it contains 7 neutrons. So the mass number is this number above the symbol that represents an atom. The lower number represents the atomic number, the number of protons. So here you have 2 carbon atoms. They contain the ID, the identifications, they contain 6 protons, but the number of neutrons can vary, and so does the mass number, because the mass number represents the number of protons plus the number of neutrons. So these 2 carbon atoms are still stable. But now let's look at carbon 14. So we talked about carbon 12 and carbon 13. Both of them have 6 protons, here represented in yellow with the plus the sign. But now look at the neutrons represented in pink. Carbon 12 has 6 neutrons. Carbon 13 has 7 neutrons. And now carbon 14 has 8 neutrons. 1, 2, 3, 4, 5, 6, 7, 8. Yes, this is déjà vu. I agree with you. But it's good to remember this, and we're going to apply it to a diagnostic tool, such as PET scan. Okay, so what's so special about this carbon 14? Well, because carbon 14 has 8 neutrons, now it becomes unstable. So carbon 14 is radioactive because one of the neutrons is going to explode, and it's going to release 1 electron and 1 proton. So now the proton now is going to be part of the nucleus. Therefore, that atom now has 7 protons. So it no longer is carbon. Now it becomes nitrogen because that neutron of this carbon 14, because it exploded, so 1 proton stays in the nucleus, and now because the nucleus now, instead of containing 6 protons, now contains 7 protons, now it becomes nitrogen, because 7 protons is the ID of nitrogen. And the electron that is expelled now can generate images. That energy can generate image in the PET scan. So the PET scans rely on these unstable atoms that release energy in the form of electron. Where does the extra proton come from? So it generates from the explosion of the extra neutron in the nucleus. So this neutron is converted into a proton that remains in the nucleus and into an electron that releases energy and can be captured in a PET scan. So you see here carbon 12, but carbon 14 now has an extra neutron that is explode into 1 proton. So now the nucleus has an extra proton, and the electron is released in the form of energy that can be captured in the PET scan. So the one in blue are the mass numbers. No, That's the atomic number. The mass number is in purple. Oh, you mean the circles. The red is the neutron and the blue is the proton. Okay, so the advantage of having isotopes now for our purpose is so that you can have a diagnostic tool, which are PET scans. Yes, the more neutrons, the more unstable it is. Okay, so you see that one can take advantage of this radioactive atom to use as a diagnostic tool in a PET scan. So PET scan is positron emission tomography. So for example, let's say you want to study the thyroid. So the thyroid is an organ in our body or a gland that concentrates iodine 1,000 times. There's 1,000 times more iodine in the thyroid than in other parts of the body. So if we inject an individual with this radioactive iodine 124, which is unstable, now the release of that energy can be used to generate a PET scan. And then we get an image of the thyroid. And here you can see the nodules that contain high levels of iodine. So these could be cancer cells or represent nodules that have high levels of this radioactive form of iodine. So that's the advantage of using isotopes. One can also use a glucose that has a form of that is radioactive, and that can give us an image of cancer. Why? Because cancer cells take a lot of glucose. So that can be used for a PET scan. Yes. So the radioactive iodine is injected to an individual that requires an exam on the thyroid. Okay, thank you for telling me. I admitted everybody now. Okay, so until now, we talked about neutral atoms in which the number of protons equals the number of electrons. But now we switch to ions. So ions are atoms in which the number of protons is not the same as the number of electrons. Therefore, they become charged. They can be positively charged or negatively charged. If they positively charged, then that means that they have more protons than electrons. And those are known as cations, positively charged ions. If they have more electrons than protons, they become negatively charged, and they're known as anions. So these cations and anions can form chemical bonds. So the question is, how does the PET scan detect radioactivity? So that energy is captured by the computer, okay? So that energy that is released from the explosion of a neutron. Remember, the neutron is exploded and is divided, let's say, into a proton that remains in the nucleus and expels an electron that is energy and that this can be captured by the computer. Okay, so these ions that are charged atoms can attract each other or be shared to form chemical bonds. So chemical bonds are dependent on charged atoms, on ions. So this is also important to form molecules. So molecules are formed by more than one atom. So there's 4 types of bonds that can form molecules. There's the covalent bonds that are the strongest, ionic bonds, hydrogen bonds, and van der Waals forces. So covalent bonds are the strongest because that means the two atoms will share a pair of electrons. So here you have an atom and here you have another atom. And they're going to, one ion and another ion, they're going to come together and they will share a pair of electrons. So that's a covalent bond. And by sharing those electrons, that bond becomes highly enriched in energy. So those covalent bonds are very useful to store energy. The covalent bonds cannot be broken by heat. They need enzymes to break those bonds. So molecules that are atoms that are ions that share electron bonds are very stable. To break apart those bonds, you need enzymes. And because they are highly enriched in energy, once you break the covalent bond, energy is released. So remember ATP, adenosine triphosphate, that is the energy source for our body. So in ATP, you have covalent bonds that are broken up by enzymes known as ATPases. By breaking up these electron bonds in ATP, that's how they release energy. So these covalent bonds can be single, double, or triple, depending on how many pairs of electrons are shared. So ionic bonds, these are weaker than covalent bonds. Why? Because the ions are not sharing electrons. They are just attracted to each other. So you have a cation and an anion. One is positive, one is negative, and they come together by an electrostatic force. So it does not involve sharing of electrons. They just come together because they have different charges. So it's an electrostatic force that maintains these ions together. So they're weaker than covalent bonds, and they don't require enzymes to break them apart. For example, if you think about sodium chloride, you heat it up and these ionic bonds break up. No, that ionic is stronger. No, ionic, the strongest is the covalent bond. Okay, now let's go to hydrogen bonds. So hydrogen bonds are a type of ionic bond that always involves hydrogen. So hydrogen is positively charged. Hydrogen is positively charged, so it's attracted to a negative ion. It can be oxygen or nitrogen. So hydrogen bonds are a type of ionic bonds, but in this case, hydrogen is always the cation, is always the positive bond. And then we have van der Waals forces. These are neutral atoms that temporarily become charged. So temporarily they attract each other. If you think about saran wrap that's attracted, that you use to seal a container, so that saran wrap is attracted and it seals the container through van der Waals forces. So van der Waals forces involve neutral atoms that temporarily become charged. Yes. I'm zoning out a little. Don't trust me. No, hydrogen, the covalent bonds are the strongest. And the hydrogen bonds are involved in water. Okay. So, okay, so let's look at covalent bonds. So here you have two hydrogen ions that are sharing one pair of electrons. So that's a single covalent bond. Here you have oxygen. It's sharing these two ions or sharing two pairs of electron ions. Sorry, two pairs of electrons. So this is a double covalent bond. And here you have a triple where two nitrogen ions are sharing three pairs of electrons. So in this case, this is the strongest bond among these three because it's sharing the greatest number of pairs of electrons. Okay, let's move on. So the ionic bonds attract each other because they have different charges. One is positive, one is negative. So let's look at sodium. So sodium, if it releases one electron, now it only has 10 electrons on the outer shell, on the valence, but it has 11 protons. So one electron is released, and now it becomes the sodium atom now becomes sodium ion because one electron is released. So it becomes charged because it has more protons, so it's positively charged. Now chlorine has 7 electrons, 7 on the valence, on the outer shell. So if it releases one electron, now it becomes negatively charged. So sodium and chloride can form ionic bonds, and now you form sodium chloride, which is the salt. Yes, all this is going to be on the exam. Everything I say and I mention and I show is going to be on the exam. Okay, so here you have an atom that becomes an ion because it loses one electron, therefore becomes positively charged. And the chlorine loses an electron, becomes negatively charged, and now they form sodium chloride. Is water. So here you have the water molecule with oxygen and two hydrogens. So this hydrogen can be positively charged and can form an ionic bond, the hydrogen ionic bond, with the oxygen of another water molecule. And this is a hydrogen bond. So ionic bonds, being that they are this type or a hydrogen bond, they are weaker than the covalent bond. Yes, that's true. So it gains. Here it loses one electron, so it becomes positively charged. Here it gains one electron, therefore it becomes negatively charged. Okay, I'm only going to reply to questions that have to do with the material that I'm covering. If you have other questions related to the exam and so on, I will answer those at the end. Okay, so here we have the water formed by linked by water molecules being linked with by hydrogen bonds. And here are the van der Waals forces. So these involve neutral atoms that become slightly charged temporarily, and therefore they attract each other temporarily. And this is the van der Waals force is the weakest kind of bond. Okay, now let's move on to chemical reactions. So these they are always bumping around, moving around, and they bump into each other and they can form molecules. And that's known as a chemical reaction when you form a molecule. So some chemical reactions are very complex. Let's say propane, which is a gas, in the presence of oxygen, it forms carbon dioxide, water, and it emits heat. In this case, light, it emits light. So you can use this in lanterns. So where you have propane gas that when mixed with oxygen forms carbon dioxide, water, and it emits light in this case. So this type of reaction, we say it's an exergonic reaction because it releases energy. And this energy can be used to warm up, like in heaters, or in lanterns in this case. Okay, so what are some of the parameters or factors that control the speed of a chemical reaction? First, the concentration. So if you have more particles involved, there's a better chance of them colliding and therefore reacting. So it depends on the concentration of the particles. Also, the particles are moving around and their size and temperature changes. So if you increase the temperature, that means the particles will move around faster and there's a better chance for them colliding. And then the energy required for the reaction. Some reactions will require more energy than others, and the orientation of the particles. So for the particles to bump into each other and to react, they need to be positioned in a certain way. Because let's say this is one particle and this is another particle, and if it bumps like this, it's not going to react. So it needs to be in a certain position. So if it bumps into the other particle, it can form a bond. So four important parameters that control the efficiency of a chemical reaction, let's say efficiency, is the concentration of particles, the speed at which the particles are moving, which can be controlled by temperature, the energy required, and the orientation of the particle. Okay, so we talked about energy. So there are two types of energy that can be used in chemical reactions: the potential energy and the kinetic energy. The potential energy is stored in covalent bonds. So if you think about ATP, or if you think about glucose, glucose has a lot of carbons that form covalent bonds. I will explain this soon. I have a slide showing this. So carbon atoms can form covalent bonds. So glucose has a lot of many covalent bonds, and that's why it's such a good molecule or source to produce energy. So potential energy is stored like in the battery, but it can be stored in molecules like ATP or glucose. So it's stored until something happens and the covalent bonds are broken apart. Kinetic energy is the energy of movement. So for example, water in a river or so on, that movement produces energy. So two forms of energy, the potential energy and the kinetic energy. The kinetic energy is available because the particles are moving most of the time. The potential energy is stored in covalent bonds. So what are the types of chemical reactions? So today, the material that we are covering has nothing to do with the lab. But I need to review this material so that it's fresh when we cover more complex material. So types of chemical reactions. Exergonic reactions are those that release energy. And this free energy can be used to form more complex molecules. So if the speed is faster, so the question here, so if the speed is faster, it's more likely to have a chemical reaction because the molecules will collide with each other. If you increase the speed, there will be more molecules or more particles colliding with each other. Intergonic synthesis. Intergonic reactions, they need energy to occur. For example, if you have amino acids that come together to form proteins, and this occurs in the ribosome, you have amino acids that come together to form a covalent bond. So to form a covalent bond, you need energy. That's an example of an intergonic reaction. So in exergonic reactions, covalent bonds are broken up and the energy is released. Intergonic reactions, to form, for example, covalent bonds, you need energy. So these are endergonic reactions. Sometimes exergonic reactions are coupled to endergonic reactions because you have the release of energy that is needed to form endergonic reactions. Well, no, to break apart covalent bonds, it releases energy. To form covalent bonds, you need energy. Other types of reactions are exchange reactions. That means that one atom is replaced by another to form a new molecule. Sometimes they are reversible, and this occurs in our body all the time, where you have two components to form a molecule, a different compound, and this goes back and forth, and then until there's an equilibrium. When there's an equilibrium, then this type of reaction stops. Okay, now this type of reactions is very important for physiology and occurs all the time in our body. Difference between endergonic and exergonic. Exergonic releases energy. Endergonic requires energy. So oxidation reduction or redox reactions, they occur all the time in our body. So here you have two compounds, A and compound B. So a compound that's rich in electrons, we say this compound is reduced. It's reduced, but it is because it's a reducing agent. It's going to donate electrons. It's able to donate electrons because it's enriched in electrons. Compound B is oxidized because it's not able to donate electrons, but it's able to receive electrons. So what happens in this oxidation reduction reaction? They are coupled. So compound A is going to release electrons, so it's becoming oxidized. And those electrons that are being released when A is being oxidized are going to be accepted by B. So B now is being reduced. So you have oxidation, the release of electrons, and the reduction, which is the adding of electrons. So these two reactions are coupled. And this happens in the Krebs cycle when we make ATP. So ATP, the source of energy, is made in the mitochondria, mostly is made in the mitochondria. So for this to occur in the mitochondria, there are all different types, very complex oxidation reduction reactions. in which one compound is going to be reduced and the other one is going to be oxidized. So this is the exchange of electrons. And this occurs in mitochondria, which are the organelles in our cells that make ATP from glucose. So glucose is essential when it's all those covalent bonds that are present in the glucose molecule. They are going to be broken and reformed. And when they're broken, they release electrons. Those electrons are going to be donated to other molecules. So this redox kind of coupling of these reactions occurs, for example, in mitochondria to make ATP from glucose. Okay, now we move on to chemical compounds. So we stopped atoms and elements, and now we're going to chemical compounds, which are more complex. So there are two types of chemical compounds. So they exist because they are reactions that form these compounds. So inorganic compounds and organic compounds. So what is the difference? Inorganic compounds usually don't contain carbon. So think about water. Water does not have carbon atoms. Therefore, it's an inorganic compound. They contain ionic bonds. That means that the atoms are charged and they are linked together by ionic bonds. So here you have some compounds that are inorganic. Oxygen, water, salts such as sodium chloride. So these don't have carbon, but here's an exception, carbon dioxide. Carbon dioxide is an inorganic compound, but it contains carbon. So these inorganic compounds, they are characterized mostly by not containing carbon atoms and by the ions being linked by ionic bonds. Of course, there are exceptions. Now, organic compounds, they're the opposite. They always contain carbon and hydrogen. So they contain mostly covalent bonds. So you immediately can see that inorganic compounds are not rich in energy because they don't contain covalent bonds. However, organic compounds that have covalent bonds, they have high energy content. And examples of organic compounds are carbohydrates, lipids, carbohydrates like glucose, sugar, lipids like fats, proteins, nucleic acids such as DNA, RNA, and ATP. Yes, not always. Forming bonds does not always requires energy. Covalent bonds do. What makes an atom radioactive? When the nucleus is unstable and one neutron is released and explodes into a proton and into an electron that emits energy. That's what a radioactive atom is. Organics are high in energy, while inorganic are not. Why? Because the inorganic don't contain covalent bonds. Remember, covalent bonds, because there's the sharing of these electrons, of pairs of electrons, the energy is stored in there. Okay, now let's talk about some inorganic compounds. So they're acids and bases and salts. So how do we define acids? So remember, these acid bases and salts, they can be dissolved in water. So if you have sodium chloride, you put it in water and it dissolves. That means the ions separate. They are no longer linked by ionic bonds. So that process is known as ionization. So what's an acid? Let's take, for example, hydrochloric acid. So remember, these are inorganic substances. They are ionized. They contain a positive ion, in this case, hydrogen, and a negative ion, in this case, chloride. So hydrochloric acid contains an ion that is positively charged, usually it's hydrogen, and an ion that is negatively charged, in this case, chloride. So acids usually contain hydrogen ions as the positive ion, and then all types of negatively charged ions. Now, bases, the negatively charged ion is hydroxide. So positively charged hydrogen in an acid, hydroxide negatively charged in the base. And then the cation can be all kinds. For example, sodium hydroxide. The negatively charged is hydroxide, and sodium is the cation, the positively charged. So what is a salt? A salt is an ionized molecule that does not contain hydroxide or hydrogen. So you see sodium chloride. Sodium is the cation, chloride is the anion. So here there's no hydroxide or hydrogen. So an acid contains hydrogen as the positive, as the cation, and the base contains hydroxide as the anion. The salt can contain any cation or anion that is not either hydrogen or hydroxide. So why is this important? This is important for the pH. So in our body, the pH is highly, highly regulated in our body. So for example, in the stomach, the gastric acid, which is the stomach fluid, is highly, highly acidic. So I explained this. There are cells in the stomach that produce hydrochloric acid. So what is hydrochloric acid? It contains hydrogen as the cation and chloride as the anion. So why is this important to have such an acidic pH in the stomach? This is so that it's easy for the enzymes to break apart the covalent bonds that links the molecules so that they can then cross the walls of the small intestine and go into the blood. Okay, now the blood and saliva, they have a pretty neutral pH. That means that the number of hydrogen ions is equivalent to the number of hydroxide ions. So pH 7 neutral means that the hydroxide ions is similar to the number of hydrogen ions is similar to the number of hydroxide ions. Okay, so the pH in the saliva and the blood is neutral because the number of hydrogen ions is equivalent to the number of hydroxide ions. In the stomach, the pH is very acidic. That means that the number of hydrogen ions is much higher than the number of hydroxide ions. So in the gastric fluid, the cells that make the walls of the stomach, they secrete hydrochloric acid, and that's what makes it very acidic. And you could ask, why is it that the cells in the stomach are not destroyed by this very acidic environment? Why? Because they have a fluid that is protective. And these molecules that protect the walls of in the stomach are known as prostaglandins, and we will talk about prostaglandins. Just remember, prostaglandins are protective molecules that protect the cells in the stomach from the acidic pH of the fluid, the gastric fluid. Okay. So to maintain these specific pHs, especially in the blood, that the pH has to be neutral, they are molecules that are known as buffers. What are buffers? Buffers are molecules that are used to maintain the pH of a fluid, of an environment. So, for example, carbonic acid works with bicarbonate to provide a buffering system to maintain the neutral pH in the blood. So these two molecules, carbonic acid and bicarbonate, work together to maintain the neutral pH in the blood. So if you think about Alka-Seltzer, if you have an acidic feeling in the stomach, you take Alka-Seltzer. Alka-Seltzer is a bicarbonate ion. That is the main component. And this bicarbonate is able to remove the excessive hydrogen ions. Remember, more hydrogen ions, more acidic. So the bicarbonate ion removes the hydrogen ions and becomes carbonic acid. And therefore, it lowers or it increases the pH. It allows the pH to become more neutral. Yes, why do people have acid reflux? Because the fluid from the stomach comes up to the esophagus, and the esophagus is not prepared to have an acidic fluid. That's why. Okay, so these two molecules, carbonic acid and bicarbonate, work together to maintain a neutral pH in the blood. So the carbonic acid can react with excess hydroxide ions to bring the pH to neutral, and the bicarbonate ion works or is able to absorb the excessive hydrogen ions to neutralize the pH. So these two work together to allow the blood to be at a neutral pH. So here you have this equation that represents this buffering system that is very, very important to maintain the pH in the blood. So you have carbonic acid and bicarbonate that work together to maintain the pH. The carbonic acid gets rid of too much hydroxide, and the bicarbonate gets rid of too much hydrogen. Okay, now let's discuss water. So we discussed acids, acids, bases, and salts. So those are types of inorganic substances. But the most abundant inorganic substance in our body is water. And water is essential in our body. First of all, it's an excellent solvent. So if you think about all kinds of molecules and even drugs that need to be dissolved in water to be absorbed. So how do you represent the concentration of a molecule in water? You can say grams per liter or micrograms per microliter, but you can also do a more fancy representation of the concentration of a compound in water or in a solvent. So you refer to molarity. So what is molarity? Molarity represents the moles of a compound, which depends on the molecular weight of that compound. Let's take glucose. So the molecular weight of glucose is 180. Why this number? Why this number represents the molecular weight of glucose? So glucose, this is the chemical formula of glucose. It has 6 carbons, 12 hydrogens and 6 oxygen. So if it has 6 carbons, because you have to multiply 12 by 6. This is the molecular mass, number of neutrons plus protons. So 6 times 12 is 72. Now hydrogen only has one proton, but since glucose has 12, so here you have 12. Oxygen has 16. So if you multiply by 6, that represents 96. So if you add up all these numbers, that comes up to 180. So it's the molecular weight, the weight of a molecule of glucose by adding up the weight of each of the atoms that are in this molecule. So the molecular weight is 180. So the molecular weight varies from molecule to molecule, depending on the atoms, the molecular weight of the atoms, the number of atoms in that molecule. So 1 mole of glucose represents the molecular weight in grams. So 1 mole of glucose is 180. So it represents 180 grams. So if you have a solution with glucose, you can represent the molarity of that solution that contains glucose. So the molarity would be in moles. So the molarity of glucose would be 180 grams per liter. So the molarity represents the molecular weight in 1 liter. So it's a fancy way of representing the concentration of a solution of a specific molecule depending on its molecular weight. Okay, let's move on. Now, what are other important properties of water that make it so useful in our body? Okay, first of all, water participates in most of the reactions that occur in our body. So some compounds are broken up into their components, and they require water. So this reaction, which is known as hydrolysis, requires water. So if you have a protein and want to break the protein up into amino acids, because the protein cannot cross the walls of the intestine to go into the blood for the amino acids to be distributed throughout the body. So you have to break up the protein into amino acids. That process is known as hydrolysis, and that process requires water. So if you think about a protein that is going to be broken up into amino acids, that's hydrolysis, because it requires water. Now, how about the opposite? If you have amino acids and you want to synthesize them into a protein, this occurs again at the ribosome. So you need energy, but you also need water. So this is dehydration synthesis. So in this case, you have the amino acids that are going to be brought together, forming what is known as a peptide bond. And that peptide bond is a covalent bond. And it is made in the ribosome. And it requires water. So dehydration synthesis. So a molecule of water is removed from these 2 components. Let's say these are 2 amino acids to form a peptide bond. So water is released. So you have hydrolysis, which is the breaking apart of a molecule. You need water for that to occur. And dehydration synthesis, the synthesis of a complex molecule from its atoms, and then the release of water. So you have water here participating on the breaking apart of a molecule or in the synthesis of a complex molecule from its components. So participates in chemical reactions. Water is very useful because it absorbs and releases heat. Remember that our body temperature is maintained at a very narrow range. 37 degrees can go up to 0.2 or -2% decimal points, but it's kept in a very narrow range. So how is that possible? Because of water. Water absorbs and releases heat. Think about a car. So the motor of the car, when it's running, it releases a lot of heat. So that's why you have water in the motor, because the water absorbs the heat that is generated by the working of the motor. If you run out of water, then the car will heat up and will stop working. Why? So the water there absorbs the heat. The same thing happens in our body. We need water to absorb the heat. And also, the release of water is used to lower the temperature. So when we sweat, the water, if that is a component of the sweat, evaporates. So the the conversion of water from liquid in the sweat to gas uses up a lot of energy. So that is used for maintaining temperature. So you have water that absorbs heat and water that releases heat when it goes from liquid to gas. So that's another two other very important properties of water that are used to maintain body temperature. Also, water is the major component of mucus that is used in all kinds of functions in the body. For example, the synovial fluid, the fluids that allow the, the articulations to move upon each other. That's like the synovial fluid that protects the articulations. So water is essential because it's an excellent solvent. It participates in these hydrolysis and dehydration synthesis reactions that occur all the time in the body. It absorbs and releases heat very slowly. That is the very important concept. So, for example, if you live in the desert where there's no water, in the daytime, it's very, very hot because you have the sun shining. At night, it becomes very, very cold. Why? Because there's no water to maintain the temperature. If you move to a place where there's water near a lake or a river or the ocean, the temperature change between daytime and nighttime is much is much less. Why? Because water is there to absorb and release the heat. So it maintains the temperature at a narrow range. And that's very important for maintaining temperature in our body. If there would be no water in our body, well, we couldn't survive, but also the temperature change would be enormous. Okay, now we move on to organic compounds, and we don't have that much time left. But organic compounds, remember, they contain carbon, and carbon is an essential atom for the organic compounds to allow the body to make carbohydrates, lipids, proteins, nucleic acids, and ATP. Carbon is the major atom. And why is it so important? Because carbon has, so in the nucleus, it has six neutrons, six protons. The neutrons here are in blue, the protons in yellow. So remember the carbon, if it's neutral, now it contains two electrons in the first shell. So the first shell is saturated with electrons. But the second shell, which could contain 8 electrons, now only contains 4. So despite the stomach being highly acidic, acid reflux occurs when things like the lower esophageal sphincter becomes, yes. So this is answering the question. So what happens basically is that the acid fluid comes up to the esophagus. It doesn't have the protection. Okay, so we're talking about carbon. So the first shell, you have 2 electrons, so it's saturated. But the second shell, which is the valence, only has 4 electrons. So since you're allowed 8 electrons in the outer shell, this means that a carbon can make covalent bonds with another carbon very easily because it can accept 4 more electrons. So the carbon backbone can be connected or linked to many functional groups, amino groups, NH2, phosphate groups, sulfidal groups, carbonyl groups, and carboxyl groups. So these functional groups give the molecule its personality. So it has carbon, but also it differs on these functional groups that are attached to the carbon. So there's 4 types or 5 types of organic compounds: lipids, carbohydrates, lipids, proteins, nucleic acids, and ATP. So I already told you here that the carbon is so important because of its structure, of the structure of the carbon atom, which only contains 4 electrons in the outer shell. So the carbon atom can react with many other carbons to form large molecules. If you think about glycogen, that is a form of storage of glucose in the muscles. That means that glycogen has many, many glucoses attached to each other via the carbon atoms. Many carbon atoms can be hydrophobic. That means they cannot be dissolved in water. Why is that important? Because think about the barriers. The plasma membrane has a lot of phospholipids that are hydrophobic, that cannot be dissolved in water. So the barriers in our body have to be made up of molecules that are hydrophobic, that are not dissolved in water. Because if they would be dissolved in water, they could not form a barrier. They would just be dissolved. They would just disappear. And carbon atoms can form many covalent bonds. So they can store a lot of energy. So carbon atoms easily react with other carbons. They form sometimes hydrophobic molecules that are essential to form membranes. And they form covalent bonds that are rich in energy. So they can store a lot of potential energy. Okay, now just to finish this, I talked about dehydration, synthesis, and hydrolysis. So this is very important for organic molecules. So if you think about sugars, fatty acids, amino acids, nucleotides, they bind to each other to form large molecules through dehydration synthesis. So sugars attach to each other to form polysaccharides, or amino acids attach to each other to form proteins. They release water. So this synthesis of these large molecules from their from their building blocks releases water. If you go the opposite direction, this is what happens in digestion. You have hydrolysis when large molecules break up into their components, and here you require water. So these 2 types of reactions we went over. And this we already talked about. So I finish here, and I will see you on Thursday on Zoom. So we covered everything until carbohydrates. Okay, any questions now about exams? I will be happy to answer. For the mock exam next week, will there be content-based questions? Okay, so for the mock exam, there's not going to be any physiology questions. The questions are only based on the steps that you need to take to answer the question. So it's sort of an exam based on the instruction of how you take the exam using ExamSoft. So it will have no questions on physiology, on the material that I'm covering. Okay, is that clear? Well, it's not going to be content based, but I'm going to give you some examples later of questions that are similar to the ones that will be on the exam. On September 11, the mock exam is just so that you go through the steps required to use ExamSoft to take the exam. Believe me, this is very important. Yes, the exam will have only multiple choice questions. You will take the exam on your computers or iPads, but in person at Assembly Hall. And the questions you won't have the ability to backtrack. So they will have to be taken in sequence. No, the exams are in person. Please don't be confused. The exams are in person. But while you are in the assembly hall, you're going to take the exam with your computer. You won't need Wi-Fi. It's going to be magical. You don't need Wi-Fi because you will be able to download your exam onto your computer. All this is going to be explained. Now it seems very confusing, but we'll go step by step. And that's why we have the mock exam, so that you have an overview of how this is going to function. You will have on September 11, you have to come to the assembly hall. It's going to be in person. The class is going to be in person on September 11. And the aim of that class is so that you know how to take the exam. I will also cover some material. It's like a lock browser. Exactly. You nailed it. I didn't hear you. For the mock exam next week, the questions aren't any content. It's just to get used to the software. Perfect. Yes. Yes. So what you'll do is you can ask me questions, but we will sign out and then you will sign on again. Lab questions, you ask the lab instructors. It doesn't, the mock exam for the lecture does not count for the grade. Lab questions, please direct them to your lab instructors and follow the lab schedule, the lecture lab schedule that is posted. Yes, all lecture exams are multiple choice. Yes.
Professor, I have a question.
Yes.
The mock exam, the syllabus is lecture 1, 2, 3.
Sorry?
For the mock exam, the syllabus will be lecture 1, 2, 3.
No. If you look at the syllabus, it doesn't say lecture 1, 2, 3. Did you look at the syllabus for the lecture?
Yes, I looked through a lot of subjects. I just lost. I'm sorry. I'll check again.
For the mock exam, there is no the mock exam is not going to be on lecture material, okay? You need to be present. You're going to learn how to use the software. The exam is going to be on September 25, and it covers lectures 1 through 8.
Thank you so much, Professor.
You're welcome. Yes. So you have many questions, many review questions that are already posted on our website. They're not multiple choice. They're for you to see if you know the material. There's many examples of multiple choice questions on sites that are available. Also at the back of your book, and also on mastering. Yes, you are going to download the ExamSoft application before the mock exam. Yes, I will give you the instructions before September 11th. Everything will be clear. Believe me. We're going to take it step by step. Okay? Any more questions? Yes. The questions, practice questions, they're not available yet. I didn't make available, but you have review questions that are already posted. I did not post yet multiple choice questions. I can put some examples of multiple choice questions so that you can see the type of questions that I ask. Okay? That's it? No more questions?
Professor, I have a personal question. So, because this is a recorded section, I don't know if you're gonna stop the recording and I can ask you.
Wait a minute. No, this is still recording. What you have to do is sign out, and then when everybody leaves, you sign on again, and then you can ask your personal question. Okay?
Okay.
Don't ask it now, please.
Okay.
Okay, bye-bye, everybody.
Thank you, Professor.